Seems a lot, doesn't it? When I talk to my undergrad students about things that happened 20 years ago, they look at me like I'm a fossil, but David is anything but a fossil. I actually tried, when I was in Texas giving a seminar a week or so ago, I tried pumping his students on, give me some dirt on Cannatella, and it was pathetically little. Either that or they knew that somehow the word would get back, and this would not be an advantage to their career. Anyway. So I have no dirt, but it's a real pleasure to have Catfish with us. That's his real name. David Cannatella's a different name. And he's going to tell us today about some really extraordinary work they've been doing on poison frogs in the near tropics. The floor is yours.
Thanks, Greg. Thanks. It is a real pleasure to be back and talking to you. As Craig was saying, 20 years really isn't that long. I can remember very distinctly sitting in museum lunch myself and meeting people and having people ask me, what is your research program about, or what are your questions? And my response was sort of, well, I work on frogs, and that's sort of the extent of it. And but somehow I've managed to make that. I feel I've managed to make that work, and I'd like to share with you today some of the things that some of my students have done really in terms of working with one particular group of frogs. And at the end, I want to say just a little bit about how this relates to, just the way people do science in general. Now, not a frog, but one of the things I've been doing on sabbatical, actually, aside from working here, is, the 200th anniversary of Darwin's birth is coming up next year. And I've been reading Darwin's autobiography and many of his letters that he wrote. And some of them are really fascinating. For example, there's a conversation that went on in a letter, a conversation by letter. Dear Wallace, this is Alfred Russel Wallace that Darwin is writing to. On Monday evening, I called on Bates, that's Henry Bates of the Amazon explorer, and put a difficulty before him which he could not answer. And as on some former occasion, his first suggestion was, you had better ask Wallace. So here's the question. My difficulty is, why are caterpillars sometimes so beautifully and artistically colored? And it's important to understand where Darwin is coming from here. He was thinking about butterflies. I mean, he goes on and says, they're butterflies, they're brightly colored. Obviously, this has something to do with attracting mates and sexual selection. Caterpillars aren't really reproducing. Why should they be brightly colored? And of course, most of us sort of know now that, a partial answer to this is basically, well, these caterpillars have some sort of defense. Predators learn that they're defended in some way, and once they try eating them, they quickly learn that it's not worth their time, and they leave them alone. But this was an area of active, thought and conversation back then. And so the word we use for this is aposematism. And so aposematic prey are those that are conspicuous by any particular sensory modality that you like. And today we're going to talk about visual sensory modalities. And they're unprofitable to the predator either because they're toxic, distasteful, difficult to secure. I'm going to use toxicity and distastefulness and defended today as if they're sort of synonyms. I realize they exactly aren't. People who work in pharmacology have a very distinct definition of toxicity, and they don't mean distasteful by it. But I'm going to just generally talk about all the toxic and distasteful as if it's sort of the same thing, realizing that it's not. So the coloration teaches a predator to avoid wasting its time on conspicuous and defended prey. And of course, there are many examples among amphibians, some frogs, some salamanders, and obviously there's a lot more examples throughout the animal kingdom of aposematism. But I wanted to talk today about one specific group of aposematic frogs, the poison frogs. And they are the family Dendrobatidae. There's about 250 species. They're in Central and South America. And many of them, as you can see, are brightly colored. And they also have alkaloid defenses in their skin. But within this family, there are many species that are not defended. They are quite palatable, and they're also fairly inconspicuous. You may not think this is inconspicuous, but when they're hopping amongst brown leaf litter in a dimly lit forest floor, it's pretty difficult to see them. And what else is interesting, and I'll say more about this, is some have actually fairly specialized diets, whereas others are generalists. Now, not that long ago, 1991 even, it was believed that within poison frogs, aposematism was uniquely derived. And the reason for that, in this a simple cladogram that was put together by Chuck Myers, is that, well, you can look and see that there are a bunch of brightly colored frogs that all have these lipophilic alkaloids in their skin. And one of Myers' collaborators, John Daly at NIH, had done a lot of work characterizing the alkaloids in the skins of these various species in these genera. And basically no one would really believe that that these complex compounds would be, could come from anywhere but synthesis by the frog itself. And so for this reason, it was thought to be a monophyletic group. One of my students, Juan Santos from Ecuador, he's actually really happy here because he just found a new species in the babies. And the other thing I'll point out is that we're in lowland rainforest, Ecuador. This is a long sleeve jacket with actually fiber-filled padding in it that somehow he manages to wear something that you would put on a nice chilly Berkeley morning out into the jungle because he doesn't like the insects. But anyway, Juan came to work with me being very interested in the phylogeny of dendrobatids, and what he did was for almost all these species here, he sequenced a couple of genes from these. And when we really got out the first tree, what we were just amazed to see was that there were clusters of the brightly colored species shown here in these gray boxes. So a nice cluster here of dendrobades and phylobades, but also clusters here, here, and even one here and a couple here. And some of these we knew from information in the literature actually did contain, some of them had been sampled for alkaloids. But others hadn't, many others hadn't. But nonetheless, the fact that it seemed that aposematism had originated not once but many times within this particular clade of frogs is pretty interesting. Now, at the same time, another group, Miguel Vinces and collaborators, published a similar sort of paper which reached basically similar conclusions about the origins of aposematism in these frogs. Well, this is pretty interesting in its own right, but it certainly raises a number of questions. Because evidence had begun to accumulate, a lot of it anecdotal, that in fact these frogs were getting their alkaloids by sequestering them from the prey that they ate. And a lot of this was, the sort of work by, not work, but just observations by people who had these frogs as pets, that over time some species looked like they lost their toxicity in captivity. And when you kept them on a diet of fruit flies for 3 or 4 years, they didn't seem to be as toxic anymore. And there were maybe 1 or 2 experiments done actually on a species, one species, that seemed to suggest that this might actually be the case. And of course, there was a lot of literature reporting alkaloids in the skins of the brightly colored species, but strangely, almost no one had looked at the inconspicuous brown species to see if there were actually alkaloids there. As a matter of fact, we could only find literature saying that 1 species of dull-colored frog had no alkaloids in it. And then there was also a little bit of literature suggesting that diet specialization had occurred in some of these brightly colored species, but again, that this was all under the assumption of a single origin of aposematism. So we became interested in understanding what was really going on with this pattern. We wanted to go beyond just getting some anecdotal data. And another one of my students, Kat Dars, took this on for a project, and she basically reasoned that, well, if if these brightly colored frogs have evolved multiple independent times, and that if they do have some sort of dietary specialization on the types of prey that actually might confer the alkaloid defense on the frogs, then we should see this occurring multiple times as well. A pretty straightforward idea. Getting the data to test it is not as easy as you might imagine. It involves going out to the field and not just looking at museum specimens, but you need to get the frogs right in the field, sacrifice them immediately, because even in the space of a few hours, they keep digesting what's in their stomachs. And so simply looking at museum specimens is going to typically bias you only toward exoskeletons and not toward other sorts of prey that the soft-bodied prey that you might find. So so she was able to get good data from about 9 species, quantify the diet from these. We also used the skins of most of these same frogs in a very simple assay using thin layer chromatography to just simply determine whether or not there are alkaloids in the skin. And this is really just a presence-absence sort of assay. And also we used data from the literature where a couple of workers had taken the characterization of alkaloids by John Daly and colleagues and tried to make some sort of composite score to try to assess something about the degree of toxicity or defense in these frogs. So we had some, a little bit of information from the literature that might be usable, and then our own data. And then a lot of data about prey. And so this real quickly just are the results of the alkaloid assays, and the gray boxes represent our data, and the white areas represent just species for which there was some data in the literature that we thought we could use with reliability. The results of the diet, again, there's nothing you need to take away from this other than the fact that there's a lot of data and a lot of numbers. Again, the gray boxes are our data, and the white boxes were data that were already in the literature. And we made a special effort to quantify our data in exactly the same way that Jan Caldwell did with the data that she collected, and she consulted with us, to make sure that we were, doing this so that data would be, we could pool the data together. And what Kat found in this so let me show what we have here. This is Juan's phylogeny with the same taxa, the brightly colored taxa with the gray boxes. Here are some, the taxa in bold are the ones that we have diet data and the alkaloid data for, so spread throughout the phylogeny. These little histograms show you for each species basically what the distribution of diet items look like. And I know this isn't really very interpretable, but for example, in this species, Cholesthepus inspiratus, you can see that in the categories of prey, they're fairly evenly distributed across here. This is much more of a generalist. This species, Epipades bilinguus, is highly specialized on ants and Isoptera. And so you get histograms that tend to look a little bit skewed, like this, this, this. And we did various sorts of analyses to sort of array these species in terms of whether or not we might consider them to be generalists or specialists. And no matter how you sort of cut the pile of species into 2 groups, it was really obvious that diet specialization occurs basically at the base of this group, Phyllobates and Dendrobates, at the base of this group, in this taxon, but not in this one. In this species here, which is not really a brightly colored species, actually did have diet specialization, but not in some species that are brightly colored. So there's not a perfect one-to-one match between diet specialization and being conspicuous. That's okay. The important question is, are these traits correlated in a phylogenetic framework? And there's some multivariate techniques that you can use developed by Mark Pegel to answer this question using the phylogeny and branch lengths. This is one question we asked, basically, well, are diet and the presence of alkaloid defense correlated in a phylogenetic framework? And then secondly, does the evolution of diet, however we quantify it, and alkaloid defense vary exactly with the phylogeny? In other words, is there phylogenetic signal, or is it independent of the phylogeny? As if selection is acting in some way such that there's no phylogenetic trace of the trait evolution left. And, well, the answer to the first question basically is, well, you are what you eat. That is, if you eat a lot of prey, if you have a specialized diet, then you actually have a really high probability of being defended by alkaloids. And again, here are a number of these diet variables, but we use the principal component analyses on all these just to sort of summarize it. And there's a fairly significant correlation with either the literature measure of alkaloid score or our simple binary measure of alkaloid score here. And most of these, with a couple of exceptions, are correlated, not surprisingly, because these are all measuring just different aspects, different perspectives on the diet. Okay, so diet is clearly correlated with having alkaloid, although it's not a one-to-one sort of correlation. In terms of phylogenetic signal, what we see that when you look at four of the 6 diet traits that we looked at actually do not appear to track the phylogeny exactly, meaning that it doesn't demonstrate they're under selection, but means changes in these traits just, you can think of it this way: the anagenesis along a branch of one of these traits doesn't really match up to the anagenesis along the same branch as measured by the DNA sequences. Okay? But, and when you look at the principal component overall, again, the diet doesn't seem to be tracking the phylogeny extremely well, whereas the 2 alkaloid measures actually track the phylogeny fairly closely, meaning there's some degree of phylogenetic signal in whether or not the different species have alkaloids or not. And so what does all this mean? Well, so presumed aposematism, and I'm going to say presumed at this point because we haven't experimentally demonstrated that that's going on yet, has evolved multiple times, and there seem to be correlated independent shifts to a specialized diet. Specialized meaning having large numbers of small prey such as ants, termites, mites, that have occurred within the poison frogs. The diet and alkaloid defense are actually strongly correlated, but it's not a perfect correlation, and that's going to prove to be interesting, I hope, in just a few minutes. And the alkaloid defense is closely tied to the phylogeny, but the overall diet is not. And this, to me, is actually, it's something we're still thinking about, or thinking about possible experiments to get at what this might mean. And if anyone has any ideas, I wouldn't mind hearing them. I mean, in general, it suggests to me that evolutionary shifts in diet are less constrained in some fashion than shifts in whether or not the animal uses alkaloid defense. So in sort of a real world sense, what might be happening? Well, the animals may be able to shift diet in a general sense fairly readily. If you're using alkaloids to defend yourself, it's sort of a hypothesis I have in the back of my head, is that if you're using alkaloids to defend yourself, you have a problem in that the alkaloids are going to be harmful to you also, typically. And the way some animals have gotten around this is they've evolved certain changes in sodium channel genes, as one example, such that the animal itself becomes immune to its own toxin. This has happened with tetrodotoxin a number of times. And so it may be that there's some evolution in genes that control ion channels. It doesn't necessarily have to be sodium channels, but something in these frogs is going on to where they don't very easily evolve resistance such that they can sequester alkaloids to use in defense, but that they have done it more than once. Now, okay, so what other questions does this, bring to mind? Well, I've talked specifically about alkaloids and diet, and how those are related. I've showed you that, these frogs are conspicuous, but we really haven't done anything to quantify that or get at some ideas about what they're doing other than that you know, in most cases we think that conspicuousness will help defend a toxic species like these poison frogs when a predator can learn that they're actually not worth wasting their time on. But other ways that conspicuousness can help is, for example, conspicuousness can defend a toxic species if it mimics another toxic species, and we know that as Mullerian mimicry. Conspicuousness might also defend a non-toxic species if it mimics a toxic species. And if you remember one of the slides I showed you, you know, some of these frogs that actually look like they're conspicuous seem not to have alkaloids. And in fact, to get into talking about parallel phenotypes, here are a couple of frogs. How many people think this is a toxic frog? Raise your hand. How many people think this is a toxic frog? I assume it's the rest of the people. Okay, we're going to see how good of a taxonomist you are. So, this is the toxic one. Okay? These two live sympatrically. How many people think this is a toxic frog? Almost always get a fit, and how many people think this is it? It's like, I've never had a case where people can actually really guess accurate, you know, consistently which one is which. So this is the toxic one. And again, these two live sympatrically, and just to make this a little bit more interesting, notice the name of the species of this one, Alkaloid sapro, is the same as the name of the species of this one, Alkaloid sapro. So these are apparently, they're two different morphs of the same species that live with different, seem to live with different models. And then how about these two? I mean, they're not that conspicuous. They both have some bright yellow, but yet one of these has alkaloids very clearly in the skin. Which one? This one? Or that one? So it's this one. So I can actually tell these apart. Okay, believe it or not, but it is sort of tough. Okay, so where do these guys live? So we're going to talk about these. Well, this species and its presumed mimic actually have a really widespread distribution across the Amazon basin. These are GAA maps, Global Amphibian Assessment, and these aren't too bad in terms of the accuracy. These were really bad, so I sort of fixed them. And you know, and the reason that they were bad is the literature reports are filled with misidentifications by herpetologists who can't tell the species apart. So this species and its mimic live up in this region, the northern part of Amazonia and Ecuador. This species and its mimic are in the southern part, and they actually have a narrow zone of overlap. Although I'd love to have a lot more data on that, but we know they overlap at least in a couple of sites, and that they have these general ranges. So fairly narrow distributions, but here a very wide distribution. Okay, so it looks like there's Bayesian mimicry going on, but let's actually see if we can figure out if that's really the case. Because actually, before this, as far as sort of I could tell, there weren't really any confirmed cases of Bayesian mimicry in frogs, and maybe one in salamanders, Dave? Is that about right? Yeah. So what might you expect to see if you were going to assess Bayesian mimicry? Well, you want to be able to measure conspicuousness of the frog, that is, in the predator's terms. What does a predator see when it sees a frog like this on the forest floor? And we also want to assess how similar these are to each other, you know, the model and the mimic, presumed model and mimic are to each other. Now, I just showed you cases where I think most people would agree that those three pairs I showed you are pretty similar, you know. But can we quantify that? A second aspect is to actually assay chemical defense, and one way we did this was using some simple injections of skin extracts with lab mice. And these weren't done to actually kill the mice, just to actually measure the amount of time that the mouse showed an irritated response to the injection. Another way was to actually do some experiments with predator learning to see if predators learn to avoid these frogs, and we used chicks for this. And also using chicks, we tested predator deception to see, in fact, are the mimics really fooling the predators? Okay, so to look at visual similarity, and this work was really, was made possible by collaboration with Molly Cummings, who does visual ecology. We use photometric readings of various patches of the frog's skin and also readings of the background light in the forest where the frogs were caught, actually caught, not in the lab. This meant bringing photometers to the field and taking these measurements, but that's what we did. And the light reflectance readings can be, without getting too technical about it, basically can be sort of analyzed into a brightness axis and a color contrast axis. And when you plot, there are about 15 to 20 individuals of each species that were plotted, but the ellipses show the 95% curves. When you plot these out in space, what you see is toxic species, toxic species, toxic species. Okay, these are not predefined groups. These are just simply array all the points in this general visual space, and it's presumed mimic. It's presumed mimic. It's presumed mimic. In other words, this is sympatric with this. This is sympatric with this. This is sympatric with this. So we would take this as good evidence that these are visually similar animals, aside from just the fact that it's sort of a no-brainer when you look at them that they look really similar. And what's particularly interesting is that when you look at these, the 3 toxic frogs are part of a clade of about 8 to 10 species, which are all toxic. The 3 non-toxic forms, there's two different species, but 2 morphs of one species. These 2 are actually sister taxa from a completely different part of the tree. As a matter of fact, the divergence between these 2 is as great as you can get within dendrobated, so it goes back 40 million years, are at this part of the tree. So not only does it seem there's a mimicry, Bayesian mimicry, but it's a sort of clade mimicry in a sense. There's a clade of these 3 morphs that are mimicking these models. Okay, what about chemical defense? Well, using the skin extracts of some of these frogs injected into mice, the response of the mouse in terms of the amount of time it displayed an irritable response was measured. This response was scaled among these 3 species, the 3 toxic species, among 3 toxic species and a non-toxic species from 0 to 1. And so Parvulus is the most toxic. The other 2, Bilinguus and Hanelli, are about roughly equal in irritability to a mouse. But then when you array these by conspicuousness, actually Bilinguus is the most conspicuous, again with an array from 0 to 1, and the other 2 are roughly equal in terms of their conspicuousness as measured by photometry. So this actually sets up a really interesting scenario where we have one species highly conspicuous but less toxic, another species highly toxic but less conspicuous, at least what we think is conspicuous to a predator. Now, we can actually test whether or not they're conspicuous to a predator. This was another part of Kat Darth's dissertation, which she did in Ecuador, using, they're not, they're big chicks, juvenile chicks, I guess. So, this experiment, you put a frog under a glass dome in an arena, and then you put a chick in, and if you ever raise chickens, you know that they investigate anything that looks like it might be edible. It goes over to the glass dome, and it looks there, and it sees there's something under it. And then when it pecks at the glass dome, you pick up the glass dome and let it sample the frog, okay? Meaning it'll grab the frog, and what does it do? Well, it'll either eat it or release it quickly, okay, if it's toxic. And so what we're trying to do is figure out how long it takes the chicks to learn, if they learn at all, what a toxic frog is. Okay, so the first trial, this is basically how long does it take the chick, the latency period to where it pecks on the glass dome, you know, and then you pick it up and it grabs a frog. So here, the first trial, all three species, and there are multiple individuals of frogs tested, all three species, Parvulus, Bilinguus, and Cannatella, almost immediately go over to a dome and peck on it. Second trial, okay, it takes about 40 seconds for any of the three species to go over, you know, to go over to that dome and peck on it when it's still got one of these frogs under it. Third trial, notice that Parvulus is starting to separate from the other two. Parvulus, remember, is the most toxic. And then by the fourth trial, the chick, after 2 minutes, never pecks on a glass dome. After 3 tastes of a Parvulus, it's had enough, okay? Doesn't want anymore. But notice with the others, the other two, which according to the mouse assay were of roughly equal toxicity, the other two show basically the same pattern, that eventually they get to a point after about 7 trials or so where they don't really want to peck on the dome anymore, they just leave it alone. But up to that time, they're going back and saying, well, maybe this one's not as bad tasting as the other one, you know, but eventually they learn. So these chicks can learn, and they can actually distinguish. I mean, they seem to sense something about the distastefulness in a way that mirrors what was shown with the mouse trials, which is good. Just to show you what, okay, now I'm going to show you the second experiment with these chicks actually involves the chicks making a choice. And so in this case, this chick has been taught by the first experiment that, you know, a Parvulus, it doesn't matter which, but a chick's been taught that either a Parvulus or a Cannatella or a Bilinguus is something that doesn't taste very good, okay? And so now what we do is, in an arena, we give the chick a choice of this plain brown cholestethus, which has no alkaloids, and then under this dome is either a Cannatella, a Bilinguus, or a Parvulus, one of these brightly colored defended frogs. And then you measure the amount of time it spends looking at one of the other dome. Let's see if this is going to work. There we go. Okay, so it goes over, pecks, pecks, pecks. It's not getting anything. So it goes over, looks at the other one, but no, I don't like this. Goes back, pecks. Nope. Second chick. Show you it's not just one chick. Pecks, 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 pecks. Nope. Not a chick. And these were filmed just to, you know, make this point. Normally we didn't have a bright light shining on the chicks while they were, you know, making their choice. But they learn, and they learn to tell apart very well what's good to eat and what's not. And so if we quantify, so here's the quantification of that. So again, Parvulus, the most toxic, score of 1, not as conspicuous, okay? Bilinguus, the most conspicuous but not as toxic. Cannatella, less conspicuous, you know, not as about as toxic as Bilinguus is, but less toxic than Parvulus. Okay, so what these graphs show is the gray bar shows how much time the chick, before learning, how much time the chick spends with the stimulus frog, the brightly colored one, as opposed to a control frog. And so the 1 here means that they'd be spending about equal amounts of time with both, okay? That's before learning, okay? So before learning, the chick maybe spends a little bit more time with the brightly colored frog, but it's not statistically different than 1. For Bilinguus, you know, maybe, again, it's not really statistically different than 1, and this one clearly not. So before learning, they'll go back and forth between the brown and brightly colored frogs. After learning, okay, when you give it a brown frog versus a Parvulus, it spends almost no time with the Parvulus, as I just showed you in the movie. It spends all its time with the brown frog. But now, if you swap out the Parvulus with the mimic, okay, and try it again, here's the mimic, it spends almost no time with the mimic. And the same thing here, okay, with the yellow spotted mimic. And even the same thing here with these which aren't that brightly colored. Now it does spend more time, but notice less than with before learning, and about the same amount of time whether it's the mimic or the model. So this, we thought, was actually pretty good evidence of Bayesian mimicry. The predators are responding to a mimic as they would respond to a model once they learn that an animal that's conspicuous like that is not very good tasting. And not only that, they respond to it in a couple of different ways. Notice in the most toxic frog, they really just avoid it essentially completely. In the most conspicuous frog, but not as toxic, they also avoid that one pretty completely. But in the species which just by serendipity is sort of, again, intermediate in toxicity and intermediate in conspicuousness, even though they've learned that it's not really good to eat, they still spend some substantial amount of time with it, okay? And this suggested to us that there's sort of a trade-off going on here, that Parvulus and Bilinguus, these two, essentially achieve more or less equal protection or comparable protection by being either very conspicuous or very toxic. But that Cannatella, this guy, is not as protected as either one, but then again, it's not as conspicuous as either one is as well. And so what these diagrams are basically summarizing what I just showed you, is that here we have relative toxicity, relative conspicuousness, and the relative degree of protection that the combination of toxicity and conspicuousness affords, which in both these cases is fairly high, as you can see here. It's just graphed in a different plane. But for Cannatella, less protection, but it's also less conspicuous and less toxic. So does this mean that Cannatella are just getting eaten up right and left in nature? Well, obviously not to a degree that would drive them extinct yet. And what we haven't looked at is obviously the role of crypsis. In other words, given that, there is some degree of conspicuousness, but that probably means the reverse is that, well, it's probably also cryptic to some degree. It's not that easy to detect in nature. So what we haven't done yet, maybe at some point in the future, are experiments to actually see how well predators distinguish a frog like this against the background of leaves compared to the more conspicuous frogs. How well is this coloration actually giving it some degree of protection by, being somewhat, not so much different than what you'd see on a leaf litter background. So there's a lot more to do here. Okay, so aposematism has evolved multiple times. Oops, I'm sorry. Bob through that. We have a clade of two closely related species that have three different morphs that essentially is a model clade in the mimicry sense, that they seem to be Bayesian mimics of a group of three close relatives which have various degrees of conspicuousness and toxicity. And between the two defendant species, there seems to be this sort of trade-off in that you can be protected by either being conspicuous, or you can be protected by being really toxic and not so conspicuous. But in either case, predators seem to learn pretty well to leave you alone. So you don't. It's not a case of having to excel or be hit the maximum along one axis, but there are different ways of achieving the same end. And then Cannatella, again, seems to be not protected as well, but there may be yet another type of trade-off depending on whether or not it's truly cryptic, and that remains to be investigated. Okay, now, again, to sort of make a transition into this third and last part, you see these sorts of patterns, really broadly distributed, mimicking something that's broadly distributed, and again, really narrowly distributed, mimicking something that's narrowly distributed. It really begs the question of, well, how do we get these sorts of geographic patterns in the first place? These are Amazonian species. Is there something about phylogeny that can tell us about the history of these frogs? And here's where I'd like to look a little bit at the history of Amazonia. So, as you all know, Amazonia has been the subject of a lot of interesting studies on phylogeography and biogeography, a number of them done early on here by Jim Patton. And many of these studies of Amazonia have focused on these sort of relatively young events. You probably know about Jürgen Haffer's Pleistocene refuge ideas about bird distribution. Looking at Pleistocene diversification and how organisms in Amazonia have diversified. And we tried to take a deeper time view of this. And there are a couple of events that are probably worth just thinking about. One is that the Andes as a mountain range is actually fairly young as mountain ranges go. And when the Andes really hit their final uplift, it appears that it's them uplifting it formally promoted the current day flow of the Amazon River from west to east as it does, whereas in the past there's a lot of evidence that there used to be a widespread Miocene flood basin which received marine incursions from the Caribbean as well as the Atlantic Ocean and perhaps even the Pacific. And there seems to be pretty good evidence for at least two of these Miocene marine incursions. And part of the evidence for this, aside from the geology, is that the Amazon River has things like freshwater dolphins, groups of teleosts that are phylogenetically clearly derived from marine groups, freshwater stingrays, which really they're just not swimming up the Amazon River and living there. The evidence indicates that they were isolated there quite some time ago. And if you look at these sorts of, this is sort of a little bit of a cartoon between some geological events and geographic areas, but the green here is the Amazon basin. And this is just to show that there were various ways historically, including this path up through Venezuela, where marine incursion could occur. And the whole point of this is that basically before about the middle Miocene, that most of what we think of as Amazonia, probably wasn't the most hospitable habitat and suitable for widespread diversification. And it's only maybe in the last 10 million years or so that a lot of Amazonia there was really open to diversification by lineages. Now, in following up on this dendrobated phylogeny, okay, so we're going to look. We looked at about 223 of the roughly 350 species. Now, not all of these are described. A lot of these species we know exist simply because we sequenced enough individuals to know that there's a lot of undescribed diversity there, at least in the neighborhood of 90 undescribed species. We constructed phylogenies, chronograms. One issue in doing these sorts of diversification studies are issues of incomplete sampling. And without getting into a lot of details, I just want to say we tried as best we could to take into account the fact that we only had about 40% of the species represented, which actually is still pretty good. But nonetheless, it is something you have to think about. Okay, and in terms of estimating ancestral ranges, we did use basically a couple of methods. One, DIVA, dispersal by cariance analysis, which is under a parsimony model, and then Rick Ree's software Lagrange, however one wants to say it, dispersal extinction, cladogenesis modeling. And the difference here is that, to summarize it really quickly, is that this is a model-based method using likelihood, and it can account for changes in the range of in the ancestral range of a species, both during anagenesis, so it takes branch lengths into account, and at cladogenesis. So, of course, then the components, you need a tree with branch lengths and some type of transition matrix between geographic areas. And to do this, we sort of defined 10 geographic areas based largely on what on sort of the major biogeographic regions, Amazonia, the Guyana Shield, Brazilian Shield, Venezuelan Highlands, different regions of the Andes, the Choco Central America region, which pretty much correspond to faunistic distributions already that we know about. And one of the first things it seems like a really simple-minded question, but a very simple question is, look, if we just assume that species during the process of diversification could move with equal frequency from one area to another, as if geography didn't matter, as if things had been diversifying so much that the phylogeny, the biogeographic patterns did not reflect anything about the actual geography. If we assume that that's a model, it basically looks like an unordered character state matrix, which it is in essence. That's one way of thinking about it. Alternatively, we could think of, well, so maybe as some people, including Darwin and others, have argued, was that Amazonia was the center of origin for a number of species, and that everything sort of radiates out from Amazonia. It's simplistic, but maybe worth plugging into a model and see if it's a better model than this one is. And then this third model is basically, well, let's just make just sort of really basic assumption that to move from one area to another, you have to incur a cost each time you move to a different area. So in essence, the further away you are, the more cost it is in a very general sense. And using Lagrange, we were able to easily reject this and this as being the worst likelihood models compared to this one. And so we use this model in the rest of the analysis that I'm going to show you. So and we compared it with DIVA. So here's part of the tree. Here's the rest of the tree. Okay, so I went back to the beginning. But just to give you an idea that as some people have reported, in general, the sorts of reconstruction patterns that we get for Lagrange versus DIVA are actually fairly similar at the tips, but the closer you get toward the base of the tree, DIVA tends to reconstruct, species that occur in very widespread areas, in areas B, C, D, E, F, G, and I in this case, and that has in part to do with the way the algorithm works. And Fred would be happy, I'm sure, to talk at length about this on his way to the airport. But anyway, but we got results with Lagrange, which we think made a good bit of intuitive sense. And actually, what this is telling us here is that the ancestral dendrobatids were the ancestral dendrobatid species was spread between the eastern Cordillera of the northern Andes and the Venezuelan highlands. And not surprisingly, a large number of the species from the most basal clay come from that particular region. The ancestor of dendrobatids is about somewhere in the neighborhood of about 40 million years old. This is a little bit more interesting. When we look at species that are found in the Amazon basin, we're able to reconstruct about 14 dispersals into the basin and about 18 out of the Amazon basin to adjacent areas. And what's particularly, I think, noteworthy here is that, so of these 14 dispersals, 11 of them, according to the chronogram, took place at only about as far back as 7.3 million years. In other words, basically after that formation of the modern Amazon basin, after those Miocene incursions that I was telling you about had receded, only then did we actually see a large number of lineages moving into this area. And before that time, there were only 3 dispersals that we can show, which seem to be actually moving into Amazonia. So what we know about the hard geology, or think we know about the hard geology, matches fairly well with what our reconstructions from the chronogram and ancestral area mapping. A second point is that when we take these 10 areas and we sort of group them just for simplicity into 4 super regions consisting of the Andes, Central America, Choco together, and this northern shield region of South America versus the Amazon basin, we see that within the Amazon basin and within the Central American Choco region, these 2 of the 4 are the regions that show a significant amount of increasing diversification of lineages. In other words, speciation is going at a rate faster than you'd expect according to a basic equal rates Markov model of diversification. And we've done these tests taking into account the fact that we haven't sampled all of the diversity, but assuming that, well, if we had all of the diversity, would our samples be good indications of what's going on? And so the Andes doesn't show this pattern of high diversification, neither does the Venezuela-Guyana Shield region, but Central America Choco shows this, and notably, it's after the closure of the Panamanian portal when species could actually move up into that region again, and also in the Amazon basin. So again, what we get from the phylogeny and the chronogram in terms of diversification of lineages mirrors what we see in the geology. And so this is one of those unreadable diagrams. But here is sort of a simplified version of the entire tree showing a time scale with the Miocene incursion. These are the same tree split out for the 4 super regions, and this is Amazonia. And so all the green patches here are movements into the Amazon basin, which I told you about, and they're all fairly recent. Well, almost all fairly recent. There's one very deep one there, but most of them are fairly recent. And to get back to my point about the models and the mimics, to make a far-fetched connection, if I can, between aposematism and mimicry and biogeography, it would seem to me a reasonable prediction would be, given the distribution of the models and mimics, that we should see the origin of the clade of model species, the estimate of that node occur before, but maybe not too far before, the origin of the mimic clade. I mean, that would be my a priori guess, and the estimates we get for the origins of these clade are these. So the model clade, the ancestor of the model clade appeared about roughly 8 million years ago, and these are the 95% confidence intervals. The mimic clade appeared about 4.6. Obviously, these 2 means aren't significantly different from each other because the 95% confidence intervals overlap, but nonetheless, they're in the right direction, and they're not, and it gives me a little bit of faith that what we think is going on with the mimicry system is again mirrored in the biogeography. Okay, so although we don't have complete sampling for this data set, incomplete taxon sampling doesn't seem to be an obvious problem. The neighborliness of the geographic areas is a pretty good predictor of where species, where radiations and diversifications are taking place. This is perhaps not too surprising, but it's clearly different than just sort of a random dispersal sort of event. The Amazonia assemblage of dendrobatids is relatively new due to immigration and diversification. And Amazonia and Choco Central America are showing this sort of increasing diversification. Let's see. And of course, it looks like the models are older than the mimics. And with that, since we're just out of time, I'll stop. Thank you.
Thank you. Okay, some folks may need to run the classes, so if you can do that, those that have to, but otherwise we'll take questions and discussion. So start with Jim.
Fish, I guess I'm rather surprised that you don't see an increased diversification in the Andes lineages, because that's the area that has had the most recent uplift and therefore change, particularly along vertical gradients.
I agree with you. I would have I mean, actually, I'm sure Jim is aware of this, but you know, if I were to ask you which area has more species of frogs in it, the Andes or the Amazon, and you'd probably say, well, the Amazon, of course. It's like the Andes has twice as many species of frogs as the Amazon does, easily. The only reason I can think of, Jim, is that, and I mean this could be the case, is that our sampling of the Andes is just so undersampled that we're just not picking up the signal, you know, that might really be there. I mean, we certainly had a lot of, you know, I think was pretty good representation all the way from Colombia, Venezuela, and Andes, Ecuador, Peru, and things peter out south of Peru.
What is the elevational, you know, range of that are batted?
Some of them get up to 4,000 meters.
That's what I thought.
But, and those are disappearing. But there's a reasonable diversification in the middle, sort of upper cloud forest. It's better in the lower transition cloud forest to lowlands. Yeah.
Most of that, I mean, the northern Andes in particular, is basically in the last 10 million
Yeah.
years.
Yeah, I was surprised by that also.
I have concerns about your experiment on toxicity and predatory.
Uh-huh.
We don't use the real predator of these species, obviously, because I don't think they are predicted by lab mice. But
No, we weren't using lab mice as predators.
but for the toxicity.
Oh, but just to measure whether alkaloids, I mean, it's sort of, it seems like a no-brainer because you can taste the frogs and they don't taste very good. You know, I mean, I tasted them and...
For you, but you don't look for the predator.
Well, exactly. So, I mean, I can tell you anecdotally that, you know, some people fed a couple of frogs to a couple of snakes, and that definitely the toxic frogs are spit out by the snakes, and the snake is very uncomfortable with that. And so, yeah, I mean, it's a good criticism in that always in these sorts of studies, what is a good predator, you know, or a real predator in nature. In nature, I'm sure there's more than one predator for many of these, and there could be some interaction of predators, or whether or not a predator is present in a particular area that could affect the whole system. So we were, you know, using the chicks, we were just trying to do the best, you know, the best we could with at least something that we know will eat the frogs and respond in a way that indicates that whether the frog is defended by that particular predator or not. Yeah.
Do you notice any differences in like escape behavior within the mimics or the models?
Absolutely.
Or between the mimics and the models?
Absolutely. As a matter of fact, these are different than, I have to go back to a particular picture to show you this. This wasn't a setup question. Tom was actually an undergraduate in my lab at UT. So in all these animals, so this is a non-toxic one. All these non-toxic ones actually have fatter muscle masses on the tibias and such compared to the toxic ones. And when you just, again, we haven't done the experiments, but you casually put them on a surface and make them run, these guys leap long distances in unexpected directions and quickly try and hide wherever they can. These guys that are toxic just sort of take these short little leaps consistently. It's like they're not too worried that you're going to, you know, yeah, you're bothering me, but I'm not that concerned about you. And it's really consistent among a lot of these. So they don't appear to mimic an escape behavior. It seems to be a mostly visual type signal.
I had a question about the dietary studies. So you found that there was a lot of plasticity in the diet in terms of which ones were specialists versus generalists, and was there any data to show that what was available in the habitat? Like, were the areas where they had specialists, were there actually huge amounts of ants available compared to other areas?
We did do some availability studies. We didn't, you know, frankly, we did enough to convince us that if we really did all the availability studies we needed to do, we'd never ever get anything done.
Yeah.
You know,
Especially in the trunk.
and so, but availability is clearly an issue because I mean, I'm not trying to sweep anything under the rug, but ants aren't the only things that have the alkaloids. It's been, Daly's work has shown that some of these frogs eat certain species of millipedes and sequester those alkaloids. Some eat certain beetles and sequester those alkaloids. So the pattern of eating lots of ants is a very common pattern, but it's not the only prey that the frogs are getting alkaloids from. And not all the frogs have the same alkaloids, and we don't even know whether there's like seasonal variation in whether a frog becomes less toxic at certain times of the year when it's not eating, you know, prey because it's dried out and there's nothing to eat. You know, there's a lot to be done with this. It's by no means, yeah.
Going back to the predator learning and the toxicity, I definitely think there might be the trade-off and the role of crypsis might be explaining the differences you see. But I was just wondering also if just simply, because the toxicity scores are sort of scaled by this one, like one, you have the most toxic one.
Right.
And then, like, so the difference in numbers seems little, but it's like, what, 10% between the other two? So might that explain the magnitude of differences?
It could. I mean, we scaled them
Like
because
there's maybe a threshold where like the
Right.
chicks like aren't that affected anymore, especially
Right,
if you
yeah.
have like a 10-15% reduction in toxicity.
yeah, certainly that's our assays of toxicity weren't in any means, you know, absolute. It wasn't like an LD50 or something,
Right.
where, you know, where you could, but that's, you know, that's, I really the point we're just trying to make is that there are a number of ways to defend yourself against a predator, and different species may be using, you know, might be using different ones.
Okay, so take one more, then I'm going to let you go.
So in these kinds of systems, frequency-dependent selection can be really important.
Yeah.
Do you have any information on the relative population